Hey Guru Nation, thank you so much for watching. Make sure you like, subscribe, comment, share. Look, today I want to talk about why I'm always saying small is the new big, especially at the site level. And for those of you who make it all the way till the end of this video, you got a special treat because yours truly is being a test dummy for our CRC who were training on drawing blood for our studies. And we had an instructor there. The instructor didn't want to be on camera, but she's a trained instructor. She's been doing this for at least a decade, if not more. So she was giving pointers on how to draw my blood. So for those of you that can make it till the end, you get to see me getting actual blood taken out of me. But why is small is the new big, especially at the site level? Everybody who wants to break into research thinks, okay, the bigger the institution, the better. Like UCLA, UCSD, Harvard. Let me go to those places. Those places are well established. Those places have very little incentive to bring you to be a study coordinator and give you responsibilities to build your repertoire of skills so you can become a generalist. They don't want that. They don't operate that way. They don't have generalists. They have people that do certain tasks and everything's streamlined. The smaller the clinic, mine is a perfect example. Katie, who you're going to see in this video, is my second hire, my second full-time hire. The more I can get her to do, the more I can train her on. Drawing blood is one of many things. Matter of fact, this was spurred up by the fact that yesterday we had a site selection visit. Our other CRC, who is a trained phlebotomist, wasn't there. So I was doing the site selection visit with a monitor. Katie was seeing a patient in the other room. And I told Katie, hey, if you need anything, pull me out. The phlebotomist should be available for you. Well, it turns out the phlebotomist wasn't. The phlebotomist got busy. And instead of making the patient wait, Katie came and got me, which is what I've trained her on. Patient centricity is what we believe in. And we don't want to make the patient wait any longer than they have to. They're already volunteering their time so generously in these studies. So she pulled me out of the SSV. I had to draw the blood, go back to the SSV. It kind of disrupts the flow of the site selection visit. Luckily, the PI covered for me and did a really good job during the site selection visit, even giving a site tour, which is usually what I do. So I said, okay, tomorrow, Katie, we're going to train you on we're going to start training you on how to draw blood. So it's my incentive for her to learn more. I did the same thing with her with e-regulatory. I'm doing the same thing with her with patient recruitment. I'm doing the same thing with her with learning how to do site feasibility. I'm doing the same thing with her with everything. Why? I, the owner, have every incentive to give her more responsibilities and make her a generalist, because the more I can delegate to her, the more time I have freed up to do whatever I need to do, which in my case is work on my YouTube channel like this, work on my CRA Academy, work on my CRC Academy, work on things outside of the clinic, or even work on building the site. Yuma Clinical Trials, shameless plug. Work on building the site, biz dev, and getting more studies and finding more doctors and things like that. So I have every incentive to get my coordinators not only up to speed, but get them to be generalists so that they can do more. There's very different incentive structures from a small clinic to a large one to a university or a hospital. It's very different. I think it benefits the employee, the individual, tremendously, especially when they're first starting out. Because what do people do when they first start out? They want to launch their careers as quickly as possible. And the best environment to do that is at a small research clinic, because you can do that quickly. I've had coordinators I brought with zero research experience. Within a year, they were CRAs. That's not typical, but it's possible because we're giving them responsibilities. So that's why I say small is the new big. Don't neglect the small research clinics in your community. Now, you're not going to always find them on job postings. They don't have HR posting things on a regular basis. but they exist. And they're out there on LinkedIn, and they're out there on Google, and they're out there in your community. And some of them, believe it or not, are actually waiting for people like you to walk in there and say, hey, this is who I am. Here's my resume. I want to get in this industry. I watched this crazy guy on YouTube, and he told me that sites need help getting patients, getting studies, getting clinicians, or building their brand. I can help you do, and then you insert whichever ones you can help them do. So that's it. That's how you approach them. That's why small is the new big, because they're more likely to say yes. Try that strategy at UCLA or Harvard or any other academic medical center. They're going to say, oh, that's nice, you can do that. But we have full departments dedicated to each of those 4 things you mentioned. And we only want people with experience because we don't want to train. We want somebody who knows what they're doing to just plug them into a system. You're going to do data entry. You're going to do regulatory. You're going to do investigational product management. You're going to do project management. You're going to do startup. This is how they think. It's streamlined. Whereas at a smaller clinic, I'm looking for potential. I'm looking for someone with a good attitude who's willing to learn and is willing to experiment things to see what they like and what they don't like. Even between my first 2 hires, Desiree and Katie, we learned that one is more operational minded and the other one is more into marketing. And so we're going to focus their career trajectories at my site on those 2 lanes. And that's just a tiny example. I mean, of course, both can do operations and both can do marketing, but we find what their affinities are. One prefers this, so we're going to go that route, as opposed to forcing somebody to do something they don't want to do, which typically happens at the larger institutions, or at least that's what the larger institutions fear is, hey, we're going to force you to do this role and you're not going to like it because you don't know what it is, because you don't have experience, and then we don't need you anymore. That's not how it works in the small sites. And the small sites we hire for potential. So anyways, perfect segue to me getting my blood drawn and training our coordinator on how to draw blood. She did a really good job. So check it out. Okay, so right now we're teaching Katie. See, this is one of the things about being a coordinator at a smaller clinic. You can do everything. Right now we're training her how to draw blood. And we have a real phlebotomy instructor in the background. You'll hear her voice. How's it going?
Hi.
How's it going, young lady?
It's going.
We respect privacy and don't put her on.
Thank you.
She calls me Crush from Finding Nemo.
So for this one, am I going up like that way?
Yes, you are. I just try to make it to go at an angle, okay?
Okay.
Yesterday Katie got me, but it did not go in the tube. So today we're trying a different. So you never want to use the same vein, right?
No, preferably not. Just so it won't blow. Does that make sense?
Mm-hmm.
Plus, it's also more comfortable for a patient not to go in the same vein, since it's already, even though
already damaged you.
Mm-hmm.
Even though it's not as bruised or bruised at all, but there's still tenderness.
Well, some patients, when they get a lot of blood draws, they get like hard veins, right?
Yeah. And it all depends. Honestly, a lot of it depends on the hydration of the patient as well.
Mm-hmm.
That's going to depend whether you bruise, whether you get repoked,
Well,
whether
I'm
there's
super
stimulation.
hydrated. I drink a lot of water, kombucha, and coffee. It's probably good. So what does Katie do? Like so far, you put rubbing alcohol.
Mm-hmm. Well, first I did the tourniquet to see where your vein was.
Okay.
And then rubbing alcohol, and now the tourniquet again.
Okay.
When you are going to poke them, grab the needle from the wing, and it'll give you more. And see, instead of grabbing it like that, grab it by the wing.
Like
there
this.
You go. And that way you can hold on to the wing and to be able to put your tube in. But another thing that I was going to mention to you is look at where your supplies are at. You want to have them to where you can reach them without going across.
Okay, I'm going to take this off of you real quick while we set it up.
So in the meantime, we could talk about Katie they're trying to there's a new strategy now. If we don't screen patients, they will do a weekly call and make us do a report until we screen a patient. It's like their form of torture.
Yeah, it sucks.
We can't name the company, but it's another strategy they're using to get more patients enrolled. So, no screening patient, report this week. As soon as we screen one, okay, no report needed this week. Good.
Perfect. Okay, you're retouching the vein, so now you gotta re-clean it.
I know, I noticed that right when I freaking was going down to it.
That's a good habit, though, because not everyone does that. So even if touching it with a glove doesn't matter.
No,
nothing
it doesn't matter because
touch.
The gloves are protecting the phlebotomist's hands, not the patient. Because even though there's gloves on there, germs get on gloves. You know, dirt, you know, whatever, micro things that we can't see. Make sure that your bevel is up. There you go.
That would be smart. Sorry, don't hurt you.
It's only going on YouTube.
Great. If it goes wrong, you can't post this.
Okay. We'll do blooper reel.
A blooper reel. So I can literally poke it just holding this. That's all the pressure I need?
Mm-hmm.
Okay. Oh God, I'm nervous. Okay.
Don't be nervous. You're gonna do fine.
Is there a way to grab it, like with the both butterflies? You know, I usually hold those things together. Like
you can. A lot of
it
people
doesn't
do.
matter.
It just depends on your comfort.
Preference.
Okay, so for his veins, do I need to pull? Because last time when I pulled it, that's when it came out.
Okay, you want to anchor it so it doesn't move. As soon as the needle's in, you can let go of that anchor.
Okay.
Go a little further down. I'm going to start right there.
Right there?
Go ahead. Swift motion, go. Don't go as slow as you're, okay, see, now it's in there. Okay, now you want to grab your tube. Okay, as soon as you put it in and there's blood return, go ahead and release the tourniquet. So, to avoid hemoconcentration. Okay, perfect.
Oh, it popped out.
It's okay. You did what you did what you had to do.
Okay. So I'm good to pull it out?
Okay, go ahead and place the, there you go.
Yeah, and with these tubes,
don't let it.
Well, that's actually a good thing because.
See, at that moment, you would have asked the patient, go ahead and put pressure. That way you can lock your needle right away.
How do I lock
because
the needle?
That's dangerous. That could be dangerous for you because you can get poked.
Okay.
Okay.
Here, will you lock your arm?
So the way to do it in that situation is
you're gonna hold on to that and then pull up. There you go.
So if that happened in real life, like on a real patient, she would say hold.
If that were to happen on a real patient, she would have drawn another tube because if since the tube fell, it got a little. So it can cause it to be hemolyzed, giving you inaccurate results.
But the good news about these ones, so this purple top, usually not centrifuged.
No, it's not.
And so these are, she actually got enough to where they could run samples.
Yeah.
Like if this was in a study, we would still use this.
if you were to
This
get
would still
it,
be usable.
yeah, even if you were to get it to this very first right here where the sticker starts or ends, that'd be good too.
Yeah, so you got
that's
plenty.
mainly used for a CBC or
hematology.
Yeah.
This one. Yeah, CBC.
So as soon as you put in your tube, as soon as you're done, grabbing your tube or whatever, make sure that you have the patient put pressure on their own arm.
Okay.
That way you can lock your needle right away. That is the first thing you want to do for your safety.
The needle.
Okay?
Good job.
Almost there.
First tube you ever got?
Yeah,
well,
you
it fell out.
You got it. You got blood. Yesterday there was no blood in the tube. Today there's blood in the tube.
If you get a little more comfortable, maybe you could do a straight needle.
No, it doesn't.
Even I would not
that's
be comfortable with
hard.
that. I've tried it a few times.
It's the best way to get the blood. It's the best blood you can get, honestly, because it's directly into the vein, and it goes from the vein straight into the tube versus traveling through the tubing.
Okay.
And that causes. See, laboratories don't really like to use butterflies because of the hemolyzation.
Yeah. And
they're expensive too.
And they're very expensive.
For the tube, I have to hold it the entire time. for it to not slip out?
No, I think you didn't push it in like all the way.
Yeah, it probably wasn't pushed all the way
because you're supposed to hear it click,
on
right?
there, yeah, once it's on there, you know, like it's on there,
Mm-hmm.
and then you have to, you know, pull it out and then put a different one in or whatever.
But that's good. That's good practice.
But you did really good. I mean, there's no buts about it. You did really good.
Practice.
Try not to be so nervous, and just safety for you. So as soon as you pull out that needle, okay, go ahead and put pressure. Lock your needle right away. Okay. That's like the most important thing you want to do. Okay.
Good job.
Yeah.
Thank you very much.
You're very welcome.
Unidentified person.